that pops up on everybody's meeting window. Um, did not see it, huh?
Let me try hitting it.
Thanks.
Well,
Was that
I'm not the host.
okay?
Yeah, it's recording.
Great. It looks like it is recording. So this meeting will start with the very typical, is it recording? 2 minutes that we have back and forth. Uh, anyway, I just wanted to introduce myself real quickly. My name's Walt Baxter. I'm the co-chair of the Industry Liaison Committee with Linda Narhi, who's also on the call. And Linda's going to be the director of activities tonight. I'm just the slide manipulator. But we are excited to bring you another fantastic career panel tonight. And this event is sponsored by a group called the Bioengineering Institute of California, which unites all 10 University of California campuses. Thank you for joining from wherever you are across our state. I've got on the screen right now a image showing our webpage, and we are the Industry Liaison Committee. If you'd like to get more information about us from the front page, you can read all about us here. And if you click on resources under the Industry Liaison Committee, excuse me, you'll see previous webinars that you're welcome to watch, patents, entrepreneurship, PhDs, and undergraduate strategies for success. And so I just want to make sure everybody has an opportunity to go see that. There's a link to this webpage in the chat. And the last thing I'll let all of the students and faculty online know is that we've saved a ton of time for Q&A today. So definitely feel free to use the chat to stack up questions as you go. Linda and I will be monitoring those. And you also could raise your hand once we get to the Q&A section, and I'll give you a shout. I'll call you out and you can unmute and ask a question. So Linda, why don't I let you get started?
Okay. So again, we're grateful for everybody that joined, and we're also very excited with the speakers that we have. And so, since this is a career panel, that will be part of it. We asked all of the 4 panelists to give us their career journey, but also because this is a topic where I know a lot of folks are interested in just how AI is currently being used. We've also asked them to present a little bit about what they're currently working on. I'll ask you to wait till we have all 4 present, and then we'll do the Q&A at the end, because also, might be a question more than one of them can answer as well. I'm not going to introduce them individually then, going to big intros because of that, because they're going to introduce themselves. I did just want to reiterate what Walt said, that we are leaving plenty of time for Q&A, and you can do it by chat, or we'd prefer if you would raise your hand and actually let us see your face when you're asking, but we understand that might not work for everybody. So we'll be monitoring both kinds of questions. So we're going to start it out with Jennitte, and I'll let her take it away.
All right, thanks, Linda. Thanks for having me. My name is Jennitte Stevens. I'm currently the Chief Technical Operations Officer at Insitro, and I'll tell you a little bit about the exciting work at Insitro in a moment. But to start, we'll go back in time to.
Sorry,
Yeah, if you want to go to full screen.
1 second here. I'm slowly getting to where you want to be. I apologize.
Yeah, no worries. So I started, I guess, my career in the sciences back at the University of Arizona, where I did my undergraduate. I was a molecular and cellular biology major. I was, really interested in DNA and genetics and how genes turned into proteins and heredity and, through my high school biology classes. So I was really drawn to that major. I think the most, really foundational, work I did though as my undergraduate, I was able to get into an undergraduate research program that was sponsored by the HHMI that allowed me to work in a laboratory part-time during the school year and full-time over the summer, and really got my hands wet working with graduate students and postdocs. I just fell in love with the lab, sequencing genes and cloning and, a lot of molecular biology. This dates me, this was pre-human genome and actually pre-automated sequencing. So it was the traditional gel pour, and like, you see the gels on old TV shows. That's what I did in my undergraduate day. But I just really fell in love with the lab. And certainly a lot of my colleagues and fellow students were considering careers and going to medical school. And I contemplated that for a little bit, but I really loved the research and decided to go to grad school. So, I applied to grad schools. I decided on UCLA, but in the summer in between my undergraduate and graduate, I did an internship at Rohm and Haas, and that really gave me the first perspective of what life was like in industry, because there wasn't really, there weren't career seminars like this when I was an undergraduate, and there was like a lot fewer opportunities, or, at least like there were less of connections between industry and academia at the time. But my advisor, knew the lab head at this company that was actually producing, protein receptors to screen insecticides at Rohm and Haas. So I was in the protein lab there. But, it got me a sense like, oh, like the work I'm doing, which was really basic biology, can actually be applied in various ways, the same skill sets. So kind of, put a little bug in the back of my head, but I went, it was a great summer on the East Coast. And then I started my journey at UCLA. And, this was a great experience. I went in through a program called Access. I still believe it's there, but a lot of larger universities have this that, rather than going into a specific lab or a specific department, you get to explore the variety of departments and have access to all of what UCLA offered in the life sciences. So I rotated through a number of labs and landed in Arnie Burke in the Molecular Biology Institute studying regulation of mammalian transcription. Really went deep, and then allowed me to, like, it was a biochemistry lab, like purifying out and looking at interactions with transcription factors. But this, we had discovered a brand new transcription factor that, was part of a larger regulatory complex that hadn't been identified before. And again, pre-human genome, so we had to sequence the protein, and then I had to kind of try to clone it out of various cells and then sequence that myself. And then because it hadn't been discovered before, I had the opportunity to actually do some genetics and make, knock out mouse and knock it out of cells, and then understood that it was a critical regulator of both viral and MAP kinase signaling. So it was a really great journey, and it really taught me, because I had to go outside the lab and collaborate with other labs to get this work done, and had a great support network from my advisor and others that allowed me to take a unique angle that actually ended up in a science paper, which was really, really exciting. And through that journey, again, I love the research. I still love the lab. Like, my grad school days are probably the fondest. Like, just the independence and the freedom to kind of work on what you want, and was great. But I also saw, was, contemplating going on to an academic career, but also, saw the time and the work and the length, the struggles of like my postdoc colleagues in the lab at the time. And I wasn't wedded to, like, I have to work just on this one thing. Like, I really liked exploring other projects and kind of working with other labs. And so as I was applying for postdocs, there was a small company in Santa Monica. It was called AmSite at the time. It was bought up by Reneron years later. I couldn't find an AmSite logo, so I grabbed the logo instead. I was working on islet cell transplants for type 1 diabetic patients. And, the job was a senior scientist to actually go in and see if you could reprogram beta cells and to reproduce for transplants. So again, very, basic research, but, still applied to a company. So I decided to take the leap into industry and took on this small company. And was there for about a year. And again, this was also, still dating myself, pre-iPSC cells, pre-stem cells. So, And, I think I just saw a paper, which is like last year, the year before that, I think it was the Mellon Lab finally cracked that problem. So it was well before its time. And, I think funding was challenging. So an opportunity for me to join Amgen happened then in the protein science department. I was like, great, I worked on protein expression in my PhD. I know all about that. So I, started my career at Amgen, first as an individual contributor, scientist in the lab. And what was, really great about being in a group in protein sciences and biologics is that it's kind of the center piece of the discovery research organization, that it supports and designs and generates all of the biologic molecules for every single program at Amgen. So it was an incredible experience because you had to become an expert in your own technology, to, making proteins, engineering proteins. And this is where I got exposure to automation and machine learning, capabilities later on as those are coming online. But then you're also working with the therapeutic areas such as oncology and metabolic disease and inflammation. And you have to connect the dots between understanding the biology of the disease, the particular pathway you want to go after, and then how do you design a drug, say an antibody or a bispecific or even a small molecule to that protein that'll have effect on the disease in patients. So it's kind of multidisciplinary, integrating all of those inputs, but then you learn the differences of what it takes to treat an oncology or a cancer versus, which is, has a different level of considerations than treating like a chronic disease such as obesity or heart disease. And so, I grew my career through there as an individual contributor to a group leader to a functional leader. And then I think it was about halfway through my journey, because I worked so closely on antibody design and development, I got to know the folks in process development, and actually Linda was one of them. And I had the opportunity to make a switch from research into operations or process development. And process development, if you don't know that term, it's really, the work and the study and the science that goes into taking those molecules that researchers make and then figuring out how to make them and deliver them to patients. And just the rigor and the science that goes behind that to ensure you have quality and control and understand your processes to scale those up to very large scales that you can do it again and again reliably and safely and to meet the regulatory requirements of patients. And this was, I thought I knew what I was getting myself into after being in research, but it was a huge learning opportunity, learning curve for me. But had an opportunity to go into the FDA and be a part of IND and marketing application filings and working with regulators and understanding those process. It was really eye-opening and stretched and helped me grow tremendously. So was really, really exciting there. So I ended up as an executive director of early stage drug substance. Again, my hands are in the pot of all the molecules. So every molecule that Amgen produced kind of went through my team, both large molecules, small molecules, cell therapies, siRNA. In addition, it's the same thing. You have to understand what the project needs, move those products forward, but then you also have to be leading the technology advancement of your teams. And so again, as advancing and bringing Amgen's cell line and process technologies to the 21st century, incorporating next-gen sequencing, image-based automation and cell sorting, and again, machine learning models, starting to incorporate there into my career to try to predict what are the best process conditions, what are the best cell lines to manufacture our biologics. And then, so mid-2024, I had the opportunity, the CSO of insitro, Philip Tagari, who was actually the head of therapeutic discovery at Amgen, who I worked closely with for years, said, "Hey, Jennitte, what are you doing?" There's this great opportunity at insitro. It's small. I mean, let's say small. It's about 250 people. Um, you know, startup that the founder Daphne Kohler, who is I think one of the early originators, Stanford professor of AI. Had this vision several years ago about the application of AI and machine learning to be applied to drug discovery. And I'll talk a little bit about that in a second. Um, you know, and these were some of the things I was starting to see at my career at Amgen. It's like, AI, machine learning gets this buzzword, but then it's actually hard to do in this space because it's so complex. If you don't have your data organized in the right way, if you don't have the right metadata, it makes it really difficult to get a meaningful output and meaningful model creation. And Daphne saw this and said, I want to create a data platform that allows us to generate data at scale that is right for machine learning insights that can find better targets and bring molecules faster to patients. So I joined insitro, and so I lead technical operations, and the groups are there below. And it's on one hand of my job, and it really mirrors my career at Amgen. So I'm leading teams that are supporting very early discovery research, but our machine learning platform. So generating data at scale, high throughput, so functional genomics, high throughput sequencing, single cell sequencing, then genomic screening. This is image-based screening, both of RAID and pooled. I lead the automation engineering team, and then also supporting our drug development through bioassays and research operations. And then I also lead process development, CMC, and we're moving our first molecules into the clinic next year. So it's a really exciting role, really exciting company. I couldn't be more thrilled to have made the jump. And if you want to go, if I'm talking too long, I move on to the next person, but I'll flip.
Yeah, I do want to say before we move on, I apologize to the speakers. I slammed these slides together and inadvertently included a Medtronic symbol in the bottom right. So just so everybody
I don't
knows.
work. I'm not associated with Medtronic.
That's all my fault, right? So I apologize for that. Thank you, Jennitte. All right, there we go.
Okay. So as I mentioned, so Daphne Kohler, our founder, kind of came up with this concept 7 years ago, I think, before people were even connecting the dots. And, we're kind of at the limit of what human scientists can do and find from data. And this is evident that, biotech companies, many of them are kind of going after the same handful of targets, and Amgen was in the same position. And you really need to unlock the deeper threads of human biology. We need the right data. We need that data in scale, and then we need the right machine learning models pointing at that data. So insitro built an incredible automation and imaging capability in the laboratories that I run that allows us to generate disease-specific models, perturb them at genome-wide scale, and then image the impact and find those insights. And then we have machine learning scientists that are looking through like UK Biobank and human genetic data that's associated with diseased imaging data that allows us to impute disease phenotypes, and then combining those two that really drive unique insights and sheds light to new biology and new pathways that have been previously undiscovered to be in that disease. And then we're applying additional tools to help us rapidly develop drugs to these novel targets. So if you want to go to the next slide. So again, we're building a causal biology platform that combines data from, the humans, clinical data, genome-wide data from human patients, coupled that with these experiments in the lab that allows us to really drive insights. And then we've built, platforms around these, so we do them in a very similar way. If you want to go to the next slide. That allows us to scale and repeatedly, and then combine experiments that's further building our data sets that's enable our AI discovery engine.
Thank you, Jennitte. That was fantastic. I'm sure we're gathering all the questions. I'm going to turn it over to Jasmine now, who actually does have a mini Medtronic symbol in the bottom right. Many apologies.
Yes, that's
Jasmine,
right.
take it away.
Yeah, sure. So my name's Jasmine. This is my background, my career path for how I got to Medtronic. So I grew up in Auckland, New Zealand. That's where I'm from and where my family is from. And at the time I wanted to be a doctor. I think a lot of people, that's kind of how they started. And I went to do a BSc, that's what it's called in New Zealand, Bachelor of Science. And I did that in biomedical sciences. And as part of that, there was a one-year thesis, and I really enjoyed that thesis. I started to really enjoy research. And I did my honors thesis in novel treatments for preeclampsia, which is a pregnancy complication. So I worked 100% in mice. And I enjoyed it so much that I decided to abandon becoming a doctor, and I went and did my PhD, also in pregnancy complications. So you can see there's a picture of me. You can actually see on my lab coat there, I have a little placenta and a little heart, because I was doing vascular research in the placenta, particularly to do with preeclampsia and gestational diabetes, which are two pregnancy complications. And again, I was 100% in mice. And after my PhD, I fulfilled a lifelong dream of mine, which was to move to the US. Pretty difficult to move to the US when you're from overseas. You need to get a special visa. But one cool way that you can move to the US is through doing research. So I got a J1 visa that allowed me to do my postdoc in the US. And at the time, I didn't want to work in mice anymore. So I technically did my postdoc at USC, but I was in the Children's Hospital of Los Angeles. That's where I primarily worked. And I did clinical diabetes research there, type 2 diabetes in teenagers. So then I was doing my postdoc for a few years, and during my postdoc, I taught myself to code. I taught myself to code in R. And then the pandemic hit. I was doing my postdoc, and just during that, I realized I really enjoyed coding. And I also really enjoyed Los Angeles, and I didn't really want to leave. And I thought, staying in academia, you quite often have to just go where the funding is, kind of move around. I wanted a more kind of stable job where I could settle down. And can you still hear me?
Oh yeah, sound great.
Okay, good. For some reason, I just got a thing in my ear that said I disconnected. So I was teaching myself to code, and I just thought, I kind of don't want to do academia anymore. I want to see, is there an industry equivalent of what I'm already doing? And I did some research, and I found out there's this thing called data science, which was almost identical to what I was doing already, but it was industry, and I would have a more stable job in that case. And I couldn't believe my luck when I was on LinkedIn and I saw this AI data science engineer job at Medtronic Diabetes. I had not heard of Medtronic, but I could see it was in diabetes, and specifically, it looked at continuous glucose monitoring, which I had done a lot of at CHLA. So I decided to kind of do a moonshot and interview for the position, even though it was a principal position, like higher than it technically would be. But I thought if I could get in the room with them and I could convince them to hire me. And I was lucky. I got hired as a senior AI and data science engineer. And I've been there for 4.5 years now. For those who don't know what Medtronic Diabetes is, it is a insulin pump and, so Medtronic is a big company, but the diabetes division, we build insulin pumps, which autonomously deliver insulin to diabetics. And also the little white thing on the right of the pump is a continuous glucose monitor. So that plugs into your arm, it literally injects into your arm, and that measures your blood sugar continuously. And then that connects through Bluetooth to your pump. And you basically have an artificial pancreas. So as you can imagine, that's super exciting. And my job is I get to crunch all the numbers behind those devices. Like you can imagine how much data you get from these devices, right? Just continuously insulin injections and glucose readings. And so I get to do all the data science on that. So on the next slide, I have kind of some idea of what we do. So of course, first of all, like how we use AI at Medtronic Diabetes, the number one is, you can imagine, is our pump algorithm, which automates the insulin delivery. So we were the first to create an automated insulin delivery system for diabetes. You can see on the top left, you could have your pump clips onto your jeans or your belt, and then there's an infusion set that goes into your abdomen that delivers the insulin. And then on the other side of the the other side of the belly, you've got a sensor that's continuously measuring your blood sugar. So of course, we have an AI algorithm that actually creates that loop of that feedback loop between the sensor and the pump of how much insulin should deliver. So that's the most obvious use of AI in our technology. I work more on the behind the scenes stuff. So on the right, I just got the post-market quality surveillance. This is a lot of what I do is, you know, when you work in a medical device company, you don't just get to release products and then forget about them. You have to constantly be monitoring how they're performing in the real world, especially when it comes to safety. And so a lot of what I do is figuring out if there's any issues in our devices. If we get a certain type of complaint, we have to go look into all the data. Is that actually happening widespread? Why could it be happening? You know, we have to take every single safety concern seriously. So, you know, I really enjoy it, honestly, because I'm constantly analyzing data and asking questions that really, you know, it really matters. You know, that's what I enjoy about it, is that this is kind of life or death. You know, insulin is absolutely necessary for these people with diabetes. Without it, they will die. They need the insulin. So we absolutely need to pay attention to it. So a lot of what I do is data analytics, surveillance, trending analysis, picking up on any abnormalities. And then I've also got on the bottom, what we've been doing more recently since 2023, I would say, in our team, which is the AI and data science team, is we do a bunch with generative AI now. Specifically, what I work on it for is LLMs that automate these quality-related processes. So for example, you know, on the bottom here, we have our tech support agents answer calls from our customers. Well, we actually use LLMs. We have our own internal LLM called Medtronic GPT, which is powered by OpenAI, but it's all in like a very, you know, closed system so that it all happens internally. We don't send any data anywhere, but it will actually bring in all that data from our tech support and also our data from our customers and automate any kind of processes we have with quality. So understanding the biggest trends of customers, of what they're calling in, understanding if there's any issues we particularly need to look at, bucketing call topics into different categories, and then being like, are we having an uptick in a certain category, that kind of thing. Because again, we are regulated by the FDA and all over the world, the different regulatory agencies. We can't let this lapse. It's very, very, you know, important that we do this. So we're always looking at how can we use large language models to kind of streamline our processes, especially when it comes to monitoring the quality of our devices. So yeah, I mean, I absolutely love it. I couldn't have dreamed up a better job for what I was looking for. I just get to work on really cool projects with AI, but also with really important and interesting data. So yeah, thank you.
Nice job, Jasmine. I think I'll pass the microphone over to Cenk, if that's okay, if you want to unmute. And Cenk, I don't know if you heard this, but I committed a faux pas. I mean, unfortunately, the Medtronic symbols are sliding with you. Those are not Cenk's. He works at Sanofi, as you'll hear in a second. My apologies.
Yeah, so I need to also put a disclaimer. I have nothing to do with Medtronic or
Good call.
disclaimer. But I have a lot of friends in Medtronic, so I really admire what they do there, and so I want to say that. So hi everybody. So this is Cenk Undey, and I'll tell my story. I'll share about my story with you. Like similar to Jasmine, so mine started also overseas in Turkey, or we call it Turkey now. So in Istanbul. So I went to Istanbul University. I'm a chemical engineer by training. So kind of coming from this area a little bit more different, maybe the direction. And I got all of my degrees from Istanbul University. I first got like interest in the material science. I wasn't sure if I want to study nanomaterials, you know, fuel cells and things like that. But then I kind of fell in love with AI and process engineering quite a bit. And once you got that virus in your system, it kind of grows very quickly. And then I was like, I want to research this much deeper and more, especially in studying, how I can apply this into manufacturing biomanufacturing sector. Maybe I'll give you a little bit more on the more like manufacturing side of things than maybe research that you heard. And then, I studied as much as I could in Istanbul. In different universities, I took some other courses because, finding AI-related courses in one university is hard. And I was all over the city. It's a big city, anybody who's there. But I had a lot of fun. Those young days were amazing. And then I took all of my research credits and PhD that I need to fulfill. And then I decided to actually learn more on industry, actually, instead of kind of staying in academics and finishing the PhD. I decided to actually go to industry and join my first pharma job in Turkey while actually carrying out my research. My idea was that I always want to come to US and do my research, but finding scholarships is not easy. And I said, okay, why don't I try applying what I learned in AI in process engineering? I want to do a lot of modeling and and AI in industry applications. And my first job was in Eczacıbaşı Pharmaceuticals. Actually, funny enough, now it's owned by Sanofi. It's kind of full circle for me. But I learned a lot about drug product or solid dosage forms and the granulation, how to make tablets, basically. So I can make it actually in my dream now. So it's very, nice, experience for me. But I applied a lot of ideas that I learned from that very first experience. It was a fantastic plant, still exists there. And then I applied while I was working in these 2 years of my time there to some scholarships abroad. But I was very purposeful and really researched a lot, like which labs that they are really doing AI in process engineering. So I can actually, study intelligent process monitoring, process control, applications and process supervision. And, like 4 or 5 schools I applied. And one of them is in Chicago. In the college is called Illinois Institute of Technology, or IIT for short. And with Professor Çınar Lab, and they were studying exactly what I really wanted to study, which is real-time knowledge-based systems and expert systems, intelligent process supervision. So, this is a day where actually we were actually sending letters to each other. There's no email. So it's amazing, how things like you should feel really, really lucky. Every month you get a letter and so on and so forth. Or facts, basically, that's all. But anyhow, so I developed a research proposal I was working on and run by him and got a scholarship from Turkish NSF and resigned my post there in the company, and then packed up and head out to Chicago for graduate school to conduct my entire PhD research there. So, and then my US journey started. At IIT in Chicago in 1998, January. And then it was amazing. Like Jennitte said, the PhD years and the postdoc years were like one of the best years I can imagine, because you get to study anything. Your life responsibilities are pretty much, your research and yourself. And that was amazing, 5 years in Chicago. But I get to study pretty much, these are the things that were not called data science at the time. It was more like computational systems or process engineering, process systems engineering. That's what we were calling them in chemical engineering. And but I had the chance to study pretty much all the algorithms you can think of that are used today, and even very advanced ones, niche ones, and anything you can think of. And to finish my PhD studies. And then I got a postdoctoral research offer from the same lab. I accepted that, and another 3 years of postdoctoral research, which is maybe perhaps the best of the first 5 years, because first 2 years where you have to defend your PhD and so on and so forth, write your thesis. It's a little bit stressful, but after that, postdoc is just some grants and research and paper writing, and exploring stuff that you don't have too much time to explore otherwise. So I did that, and we wrote a book, published a lot. and got research grants. And also I consulted with the industry in Chicago with Abbott Labs at the time. So, but about that time, I wasn't sure, should I continue with academia and in the industry? But my heart was in industry to really implement those things that I got heavily trained on in real-time intelligent, systems. About that time, an offer or opportunity came from Amgen. Amgen, this is 2003. Amgen, about a year ago from that time, acquired Immunex with the Embrel franchise. It was a huge acquisition, and it was growing. And the Amgen Rhode Island site was growing in process development that Jennitte explained the function. But this was more like process engineering, manufacturing science technology group. And they were looking for somebody who can do more like, advanced monitoring and modeling and the production support type engineer. And I got that job, and I enjoy so much from that because, it's a very heavy paced environment where you actually get to learn how you make these molecules and at scale. And we are talking about, huge bioreactors, like 16,000 liters working volume in 20,000 liters. I mean, huge, right? And then the, big, stainless steel plant that we built there and made it operational. And I was in charge of establishing, process monitoring systems and also ensuring that troubleshoot and process improvement, studies are made to make more of Embrel, which we were not making enough fast enough to. There's so much demand. So I learned a lot of my biopharma, expertise in those years, in the first 10 years I spent there. And then there was an Amgen full transformational, full potential program called is a big transformation was going on. And I was asked to do two things in my career, which is interesting points for me. Is but in Rhode Island, I had a chance to implement some of the real-time process monitoring technologies using kind of advanced statistical methods. Sometimes they are called AI. It's not exactly AI, but it's a very advanced method to detect issues or improve processes type technology. So that got a lot of awards and attention internally and externally. We got a CIO 100 award, which is an international award, innovation in healthcare. And then a lot of attentions were on us. And then I was asked to actually take a new role, come to the Thousand Oaks, which is the headquarters, to do two things. One is build a new team, more like a computational team, like the future of process development way of working, more computational versus wet experiments. How can we do that? So I had a chance to build that team. It's still in place. I'm so glad that they are, growing the capability. And then also lead this transformational program from the data and digital side to implement new technologies. So did that. In addition to that, they said, okay, since you are not busy enough, let's also consider doing an MBA, because you are very deep technically engineer, but you'd also learn the business side of things. I share this because it's also kind of interesting sometimes, what career opportunities is thrown at you. So I also did a MBA in UCLA, so go Bruins. So I'm a UCLA Anderson graduate. I learned a lot from that aspect. And then, we had a lot of opportunities to implement like significant AI, machine learning type applications, and a lot of publications to go with it. And that overall, I would say, I did some calculation. We were making some presentations to management. Over a decade, all of these, computational, digital, AI, machine learning type benefits to the business amounts to close to 100 million returns. If you had everything, all the manufacturing implementations and the lot savings and the yield improvements, if you had everything. So and then I had an opportunity to work with I wanted to get some more experience in the European type maybe company and operations, and also cell and gene therapy that I wanted to learn more. And the opportunity came with Genentech and Roche, so I kind of decided to take that. This is about like right after COVID. I led the digital team, the data and digital team in process pharmaceutical technical development globally. So got a chance to develop digital strategies for cell and gene and the other conventional modalities. And then more recently, so I have a family, and I also want to come back to the East Coast in the school, and my daughter, and starting high school, we came back to East Coast, and I started Sanofi early this year. My role is I'm heading up ICMC, or Integrated CMC Digital Transformation team. It's a transversal team. So what excites me about this role is that I get to learn vaccines, which I didn't have a chance before. So now I'm learning more about that development and manufacturing. And then the rest is pretty much what I'm already familiar with. But get to serve globally and also transversal, like vaccines, mRNA, mammalian platform, and manufacturing science and technology and CMC. So I get to, and synthetics. So all of these are very exciting to connect all the end-to-end data flow, making the data ready for AI ready operations, and then supporting teams that are doing modeling and AI. Our overall objective is to make the labs lights out labs, basically, lab in the loop approach to use more modeling and do fewer experiments. So that's kind of the I guess the trajectory over time, where I started. And maybe quickly next click to this is the plant actually I started my career. It used to be a Turkish-owned pharma eczacı-owned. I've been into every single building here and all the manufacturing spots. We have a penicillin cephalosporin units on the left, which are supposed to be separate, and then other tablet forms on the right. But then Sanofi took over, and right now it's owned by the company that I work today. So it's kind of, I always found that interesting, because this is where you started, and now I'm working in the same company. So it's kind of find it amusing. Next, very quickly. One more. So yeah, this is the kind of education I already mentioned a little bit, but one thing that I never stopped doing is that I continued my publications, and I never, abandoned the research or tracking research and what's going in my field. It's very valuable to me. I cannot explain how important it is to this day. We published this book, Batch Fermentation. It's still used actually quite a bit in education. And there's this simulator we have written and published for penicillin. It's became a benchmark, and it received a lot of citations. I think it's getting close to 1,000 now. But we published in 2003, and also UCLA Anderson with my little girl. No little anymore, but it's a little bit of that memory. And I think I covered quite a bit earlier on this. Next one, the last one, perhaps. That's my advisor on the picture. Yeah, so we have a patent at Amgen using, these are my example of my publications, and this is what I guess is very dear to my heart. Probably down the road, I'll go back to, maybe part-time teaching sometimes, after I retire. But all of these are examples of how we have applied AI and machine learning and the predictive technologies. One of them is a US patent we just got last year, provisional patent. While I was at Amgen, we developed this predicting product stability using machine learning. So it was a novel method. Typically, it's a conventional ways of doing it is available in the industry, but we said, okay, how about we try something different? It gave us very good, actually, results. And then others, are like using image processing for clonal selection. Actually, this is something, if I remember correctly, with the Jennitte team, we collaborated while I was at Amgen. It's got published recently. And then the rest are more like, how do you control cell culture while producing the protein and improve through more intelligent control. And that boosts the outcomes more significantly. So these are some examples, but you can look me up, and there's more out there if you are curious about specifics. Thank you.
Thank you, Cenk. I think at this point we'll have Jason share his screen. He's got his slides queued up. If you've got questions for our panelists, now's a great time to throw those in the chat. Linda and I will pluck them out from wherever you put them. Break for Q&A in just a second. Jason, go right ahead. Looks great.
So much for inviting. I'm a recent professor at UCLA Bioengineering. Recent as in I moved to LA less than 1 week ago. So very new and gets into the LA sphere, but I'm as you can see, I've been in the UC system for a while, but very excited to talk about my career path. But I guess I'll do it in kind of a reverse way. I'll talk about what I do in terms of research as a new professor and how that relates to AI. So as an academic, I look a lot in terms of what we can discover, and in terms of biological discovery, what a lot of times you do in research is that you find a gene or you find an interesting protein, and you go from DNA, from the genetics, to understanding what that encodes in terms of the amino acid sequence, and then with that you figure out what kind of protein it does and what kind of biological function does that protein do. And so a lot of times we look into nature and try to understand kind of from going from the DNA to the protein to the folded protein, how does cells, for instance, organize to be able to do so many different functions inside our bodies. But kind of on the flip side is how do we then do design? And so how do we, let's say you want to make a protein or make a molecule for a particular function. So there's a desired function you start off with. So let's say I want to create this next best cancer drug. And then how do you go from desired function back to identifying a molecule, or trying to design a molecule that can do that particular function. So we go from this protein that we think can do XYZ desired function, figure out the best amino acid sequence, and then we figure out the best DNA sequence that we can order in order to test whether we can make the next best cancer drug, for instance. And so a lot of AI has been embedded lately in terms of molecular design, both in terms of predicting molecular structures, but also generative AI in building new molecules as well. And so it's in line with this overall idea of, can we use AI to build the next line of drugs? And so what I specifically work on is doing AI programs, developing them and utilizing them to build proteins. And these could solve a lot of these modern day challenges. And nature has been developed throughout millions of years in order to, adapt to its ever-changing environment. But our modern problems are changing more rapidly than millions of years. For instance, climate change, cancer, viral mutations. These are happening rapidly, and so nature a lot of times can't keep up. And so in order to solve these modern problems, we also need to have modern ways to, modern methods as well. And so these AI designed proteins, which I don't do all of this, but has been shown to be useful in making medicines, for instance, making vaccines. So there is this FDA approved protein-based vaccine that's been utilized in various different countries around the world for COVID-19. Building new technology, something really kind of far in the future. What if we can use proteins to build the next silicon chips, for instance, or building these optical fibers? Now we have this very precise angstrom level of specificity or and precision, essentially. And also sustainability, such as can we build enzymes, for instance, that degrade plastics, that could do artificial photosynthesis, for instance. And so a lot of the programs that we do, and I'll talk a little bit more about how these AI programs in general work. We build these generative AI models that can design these amino acids and proteins, therefore proteins in 3D space, to build things such as nanopores that can help in terms of DNA and protein sequencing. Let's say you want to target a particular biological biomarker that's really relevant in, let's say, diabetes or in cancer. You can ask the AI program to generate a hypothesized protein that can fit into a particular region of the target. And so there's a lot of things that we could do with these AI-based programs for designing proteins. And so to get a little bit more into detail of how these AI programs work, some of you may have heard about the program called AlphaFold, which is utilizing AI to predict protein structures, which was trained on the vast database on the PDB. And so AlphaFold essentially kind of learns how does a protein fold. And so that is, in one sense, giving it a sequence and asking it to produce a predicted structure. But now we're kind of doing the reverse. And in this case, we're trying to ask the program to create a protein or something that looks like a protein to bind for a particular target, or to bind to a small molecule to create an enzyme. And so how do we do this? So we use lately these methods called diffusive methods, where they're utilized a lot in image generation, for instance. So there's these image generation models such as DALL-E, maybe you've heard of these, where you ask the DALL-E program, can you generate me a picture of a cat? And so what happens is that it starts off with randomized pixels, and based on its AI-based training on millions, billions of different images from the web, and guided by the user entered prompt, it would denoise iteratively until it creates a picture that looks like what the user wanted. And so utilizing those kind of ideas to protein design, what we do is we start off with, here is represented the different amino acids in 3D space. It starts off as complete Gaussian noise. And through its deep learning training based on, for instance, essentially AlphaFold, what AlphaFold has learned in terms of how do proteins fold, it would iteratively denoise to create something that looks like what the user wanted, essentially. And so utilizing these methods where now we understand how protein structures overall learned, or the AI programs overall learn how these AI or how these proteins fold, and then do on the flip side of designing novel proteins for the user inputted interface. So that's the research that I've been involved with, and mostly utilizing these to really understand and make molecular tools to understand cancer biology, for instance, and that's what my lab's going to be about. And in terms of my journey, I'm probably the least international out of the group. I'm all, my journey has all been in the US. I'm originally from the East Coast. I was born in Maryland. I grew up in Virginia, in this town called Virginia Beach. And when I was 18, which was at the time of Tumblr, and all these sepia toned images of In-N-Out and palm trees of California, I was very intrigued about going to California. And I did my undergrad at Berkeley in bioengineering, where I worked in various different labs at Berkeley, such as some in plant biology, some in looking into the structures of biomass as they get dissolved into biofuels. And I worked in, for instance, biomaterial research as well. All very interesting. I learned what I liked and what I didn't like. And then I went to UCSD. Well, actually, in between my bachelor's and PhD, I did work in a startup in the Bay Area for 6 months, working on integrated chip technologies for diagnostics. But I learned that quickly while working in the startup that industry was not really my cup of tea personally. I didn't really want to work on someone else's dream. I want to work on my own dream. So I decided to continue in the academic journey and doing a PhD at UCSD, where I use more rudimentary protein engineering methods for developing molecular tools to understand cellular signaling. It was a nice pairing of both bioengineering, but also to understand basic biology as well. So creating technology, but also utilizing those technologies to help inform us about, how does biology work in general. And then after my PhD, I went to Seattle to do a postdoc at the University of Washington, where I was very intrigued by these latest AI-based programs for designing proteins, thinking that is, the future of protein engineering as well. It was actually a very interesting time when I started my postdoc. It was January 2021, and that's when AlphaFold 2 was just coming to prominence. And you can see quickly that within 6 months, the programs that we use to design proteins quickly changed to more AI-based programs. And so it was kind of funny because from the lab I was working in, we'll publish papers, but then those papers were already considered old tech by the time they were published, even though the publication time took 6 months. So things were moving so rapidly that it was so hard to figure out, even within the lab, what is the latest program to use and which one is to use for which problem. But it was a very exciting time to be in during my time at University of Washington and learning and seeing the AI boom in terms of for proteins. And then just recently, less than a week ago, I'm starting my new lab at UCLA, where I'm continuing to do integrating AI into designing proteins to understand biology as well. Thanks.
Great. So you can see we have people who reflected all sorts of different applications of AI to biotech, medtech, and then academia. And hopefully that will give us a good broad basis for answering any questions. So I think first we'll say, are there any students who want to raise their hand and ask a question that way? No one ever wants to go first, right? So I'll start with a question that actually I got from a student who was not able to come, but is planning on watching the recording, which is they were actually just wondering if there's areas in drug development or device development that you think that AI is not being applied to yet, but will be really important in the future.
I don't know of any area it's not being applied to anymore. I do think, I think there's examples, insitro got into this space in terms of like trying to dissect disease biology for target discovery. I think Jason's example on, AlphaFold and predicting proteins is being applied to also, identifying therapeutic molecules, both large molecules and small molecules. Cenk's work on, taking process data and, improving insights and speed of process development. And I know there's a lot of work, certainly at Amgen and others, like trying to predict then who's the right patients to deliver our drugs to, right? What is the right patient population to speed clinical trials and improve the success rates of our molecules. So certainly, like and it's been rapid. The change has just been, in the past, I would say 5 years, especially. I think Cenk said it well, that just the impact that it's having in the places it can be applied, and it's only just going to get more and more impactful. That's my perspective. I don't know if others have.
I can say a bit on the device, medical device side. I will say that there's been, especially like when we talk about insulin therapy, there's been less of an appetite of putting AI algorithms into devices because it's seen as much more trustworthy if you have a rules-based algorithm where you absolutely understand exactly how it works. So, you know, we've proposed different ways that we could use more of the more modern AI technologies that have come out in the last few years. But there's still ways to go before, I'd say, regulators are comfortable with that, especially when it comes to transparency of the model, understanding interpretability of the model, understanding why it's making the decisions that it's making. So there's still, I think, a ways to go to build that trust of like, can we understand why a model is making the decisions that it's making, especially when it comes to kind of a life or death situation with no human in the loop, something like an insulin pump making decisions about, have you had a meal, for example? Like right now with insulin pumps, you have to tell the insulin pump that you've had a meal. We want to get to a point where, you know, could AI be used to understand that instead? But we would have to really trust that it can do that extremely well. And right now, I think there's still that appetite for, we would prefer rules-based algorithms where we totally understand every single step of what it's doing. So I think that's AI and those kind of models are gonna become more important as we learn to trust them more and more in the future.
Thanks, Jasmine. We got a couple hands up. Maybe we'll have Jason Vu unmute himself and ask a question. Jason, could you tell us where you're from first and what you're up to? Thanks.
Yeah. Hi everyone. My name is Jason. I know I'm the second Jason here, but I'm from UC Santa Cruz. I'm currently a senior bioinformatics undergraduate doing machine learning and population genomics. And so my question was in specific to Jason. I was wondering for protein design, what kind of graduate student or are you sort of looking for? Are you sort of looking for someone who can apply machine learning in the sense of predicting like something similar to AlphaFold, or would you also be interested in someone wanting to apply machine learning in order to predict health outcomes from like the recent, because from like recent protein vaccines happening, for example? Like what kind of graduate students are you sort of looking for? More wet lab, dry lab, sort of anywhere?
Yeah, and we can talk also offline as well. I think my email's provided. But in terms of what I'm personally interested in is more kind of, so here's my overall thinking is that there is huge interest in doing AI for drug design. And industry is gonna really pounce on that. Billions of dollars gonna be put into that. I'm just a starting lab. I don't think I can really compete with billions of dollars in industry and making the next line of therapeutics. But I think where there is a lot of opportunity is building AI or using AI to build proteins to understand biology. And so building different molecular tools to understand how cells operate, or how does cancer cells become a cancer cell, or how do cancer cells become drug resistant, for instance. And so in terms of the AI, it's mostly going to be utilizing the existing programs, fine-tuning them for particular chemical moieties, and trying to understand why a lot of our proteins that we design don't work as well. Because while it sounds really great, and AI has really accelerated the development of all things that we could never have thought about before, its success rate is really low as well. So clearly there's a lot that we have learned and gained from AI, but there also clearly is a lot we still have to learn as well. And so one kind of idea is that for proteins or molecules in general, there's a lot of molecular dynamics. Trying to understand not only look at proteins in terms of static structure, but also how it moves in 3D space. And so maybe adding more layers of data into protein structure predictions and that are not only based on one snapshot, but more dynamic snapshots can perhaps be able to better predict these structures and then better design proteins as well. In terms of like health outcomes, I know that is a very big goal for a lot of companies in trying to make these AI-based virtual cell models, for instance. You know, the Meta is working on this. OpenAI is working on this. Like, there's so many different companies working on it. I still believe, in my personal opinion, that we're just not there yet. We need to actually have a lot more scientists to do the hard work of getting accurate and useful data before we can make any virtual cell model. But that is one way I'm thinking about in terms of how eventually we could perhaps predict health outcomes with AI, is to be able to model the cell, or be able to model a mouse, for instance, or a human, and then be understanding all the different parameters and all the different molecular details, and then feed in, let's say, an unknown chemical and see how does the model predict whether this chemical will, cure neurodegeneration or, reverse cancer, for instance. There's also a lot of other the kind of models of where using health data I've heard about, where training AI models based on health data. Can't really comment on that, and I'm not gonna. That's not my expertise, and I'm not sure how useful those will be. But for me, I'm thinking more in terms of molecular details and using AI models to predict, molecular details.
Excellent. Hey, thanks, Jason, for the question. That was great. We'll let you connect with Jason, Dr. Zhang offline.
Okay.
I see Dr. Earthman from UCI. Jim Earthman has logged in. If you'd unmute yourself, Jim.
Yeah, well, thank you. First, want to apologize for the gratuitous use of the UCI logo, but this is maybe the only chance I've got to use this this background. I have never used it before, and I thought maybe it'd be appropriate here. But anyway, thanks very much for all the discussions. Have been very good and really interesting. I'm of course at UCI, but I'm in material science and engineering, but I'm also in biomedical engineering. I'm a co-founder of a company called Parametrics, which is now based in Washington, Seattle, Washington. AI is great, but for deep learning, you generally have to have just tons of data. And I'm just wondering if any of the panelists here have run into that problem of just not being able to get the data they need. Maybe it's proprietary data held by somebody else. It's not publicly available, or maybe just too difficult to generate. So I'm just wondering if that's becoming a bottleneck for anybody using AI.
Maybe I'll share my experiences. I had the very problem I dealt with, and actually, sometimes innovation is actually coming from a necessity. And one thing that we were dealing with is that we need to come up with some predictive models for monitoring complex manufacturing processes. Typically, how we do it is the abundance of data available. You create a model. It could be any kind of model. It could be some statistical or partial least squares, or it could be some more convolutional neural network type model or random forest, whatever, right? So, but you need some kind of historical data to do that, train the model. But in our case, we only had maybe one or two historical campaign data available, and no historical enough bases and so on and so forth. But we still wanted to achieve the same outcome versus having to wait for a year to get the data generated. And how do we solve that problem came up, and we thought hard about it. And we said, okay, what if we actually approach this a little bit more like innovative way, if you will? How do we still achieve this kind of modeling with limited data? And what we ended up doing, actually, we use some molecules that we have had experience, using those as proxies, if you will, because there's some similarity. I mean, you can still argue this, you still have historical data, but it's not exactly from the same molecule that we needed. We didn't have that molecule data. These were MABs. And but then we were able to actually create some synthetic data and some first principles data as a starting point to solve that limited data problem. and build some kind of a learning algorithm so that, hey, over time, as we generate more data, we kind of replace this synthetic data with the real data. And over time, in a couple of years, with a slow moving data accumulation, that we have a model working on. So we published like 2 refereed journal articles on the topic in the approach. I can share, or we can look it up with my name. But
Yeah,
that comes up quite a bit. Maybe one last other example I want to give. This was a manufacturing monitoring supervision type example, but other approach we are doing actually in my current job, we do, and all of my biopharma jobs, we use the same method, which is actually using, I want to maybe mention that because now that I was thinking, what else people could be using but not use enough question, also related. A AI-based modeling is kind of empirical, right? Meaning, you have data input-output modeling going on. And you need a lot of data, like the cat example, right? So you need a lot of cat pictures to get there. But we don't have huge big data actually in biopharma typically. I know Jennitte's company is trying to do that, and they're doing great. But typically, in this typical case, the size of the data sets are relatively small. But we want more explanation power of the data. It's not about the amount of data, but actually the richness of data matters for us. And to do that, to enrich the data, we actually develop a lot of what's called in silico models, more first principle based models. And we use metabolic engineering, metabolic models, for example, for cell culture, or for purification, we develop some, isotherms based models, or or medical devices, right? So we use some physics models. But those are more science-driven, or ordinary partial differential equations type things that actually are very predictive. So we use those as kind of a mini digital twins to extrapolate and churn out a lot of data through parameterization.
So
that actually gives it a lot.
So do you ever play these models against each other to see if they are consistent with each other, to give you better confidence that they're accurate?
Yes, we do that. We actually develop what's called the model competition algorithm. So I love that, because then you let them loose, and I have even tried that with our teams in multi-agent framework, so that actually those are competing against each other, and then certain criteria is fulfilled, and the winners are kind of surfacing, and you use them in automated fashion, actually. So those are the things, in my opinion, are enriching the AI machine learning applications versus, all we do is data-based approach. I think combination of this hybrid modeling is super powerful, I have to say.
I think so too. Thanks very much.
Yeah, thank you.
Sorry.
Hey, Linda.
So I think, I just realized I was muted. So Jennitte, I think you probably have something to contribute to the comment too. Do you want to provide your perspective?
Okay. Yeah, I just saw other hands up, but
yeah, and then we'll go to.
I completely agree with Cenk, and even with trying to print and generate a ton of data, like we're never, like, it's going to be a while before we have all the data, if ever, right? And so, using tools as he described, we use a lot of imputation that gives us insights based on actually kind of modeling and prediction, and kind of driving assumptions through the data, and then setting very specific experiments to kind of fill out around that space. So, it's part of where we are today, certainly, with it. And as the data keeps growing, and then actually the sophistications of large language models, I think, layered on top of this, right, that can scour the internet of data to help kind of pull things in. That wasn't a thing even just a few years ago. And I think that is also helping us enrich, the lack of data through, various sources as well. So.
Thank you.
So I'm going to call loose for a second, but this is actually partly my own curiosity, but are there industry consortiums where people are working on sharing their data so you can enlarge the data set that way?
Absolutely. maybe I'll give a couple of examples. So I think most companies are part of, but we are part of Nimble. It's a MIT consortia, MIT. I'm sorry, the University of Delaware actually runs it, but
Yeah, it's
there's a
NIST
lot of
through NIST, right?
through NIST, but Nimble has a multiple work streams where we share data, we share approaches, and the reason I thought about MIT because they are involved in providing modeling, actually, to Jim's question earlier, to share with industry more like an open source. So somebody can take it as a base model and configure for their specific molecule, which is proprietary. So there's Pistoia Alliance is another one. There's quite a few actually consortia out there that's actually enabling sharing.
Okay, so Lutz, do you want to unmute yourself and ask your question?
Yeah, hi, hello. I'm Luz. I'm from UC Berkeley. I have a question specifically for Jasmine. I am soon to be graduating senior, and I have been very interested in data analytics, and it's so interesting to me that I could see like AI usage being used for these data analytics. And I know that you mentioned that you preferred being in LA, and I think now I'm just, I guess, debating on how, I guess, you navigated breaking into this, I would say, fairly like new sector in specifically the biotech industry of using AI, specifically looking within SoCal. How was your journey? Or like your mind process through that? Like, were you willing to like, okay, if I didn't find anything else, I'm gonna go somewhere?
Yeah, honestly, there was a lot of luck involved in my situation where I just looked for jobs in LA, and I couldn't believe it really, because it was this diabetes company and they wanted a data scientist, and that's the exact area I wanted to be in. And I couldn't have been a data scientist anywhere else because I had no formal data science training, none. I was completely self-taught. I mean, I had a PhD and I'd done statistics. So in the end, like that's basically what it is anyway. But I was only able to convince them to give me a job because I had already done something so similar. So in my case, it was super lucky. I know during that time, COVID hit, there were a lot of remote opportunities. So we do have a lot of remote people as well. So we have people even in Hawaii, all over the country that work for our company. As you probably know, that's a lot less common now since COVID has they've kind of moved on from the COVID era. So I do think that it is becoming less likely to be able to get a remote job, but they still are out there. I do think where you live is important. When you're young, though, you can always try different places. It's much harder once you have a mortgage and all those things. You kind of get stuck. So I would say when you're young enough to explore, you might as well explore. You don't need to get bogged down in one place because you don't even necessarily know what you like yet. But then, yeah, I think, California is going to be one of the best places to work in biotech. So I think no matter what, there'll be somewhere in California that you'll find something you like. Yeah.
Yeah, thank you so much for sharing that. I totally agree about the California part. But yeah, thank you so much for sharing your insight.
Thank you.
I'll just add, I spent my entire career up until just last year in the Los Angeles area as well, between UCLA and Amgen. And I still live in Thousand Oaks and commute up, and it is possible.
Oh my gosh. So you're taking the 118.
Yeah.
And like, oh my gosh, you're going through that 118 to the 405 and then all the way up.
Yes.
Oh my gosh.
I also vote for California, 10 years there. But there's a lot of opportunities in the East Coast. Don't forget about Boston area.
Okay, you're right.
That's
you're
true.
Right.
Yeah, I came out to California on my honeymoon and never went back, but my daughter now lives in Boston, so there you go. So let's see, do you have any other questions? I'm going to ask one more then, because I think we have time for one more question. One of the areas that I know a lot of us are trying to move away from is using animals for model systems. And I think that's one where I've heard a lot of hype about, being able to mine human data to come up with like a computer model system that might be better at some point. Do you guys have any thoughts on that?
I think it's a grand idea. We're not there yet. But, I do think as our data gets richer and, you start to apply those models and learning back, hopefully we can get to a place, but I think, as Jasmine mentioned, when it comes to patient safety, I think the bar is much higher, right? In ensuring that, we have a good understanding of what the model is going to predict and then buy-in from the regulators as well. And so I think that bar is pretty high in my opinion. But, I do think, again, just like everything we're talking about, it can help provide insights, maybe reduce the number of animals or the numbers of studies you have to do. But I still think there needs to be that data in the loop, at least for the foreseeable future. But
I couldn't agree more. So I actually, it breaks my heart, that part of our business, that
yeah.
we should find a way to reduce. Elimination is a big bar, high bar, but and whoever has, especially if you have a pet, much deeper, right? So when you look in the eye, I have a dog, and it's very hard to think about that, but it's for the, I guess, best of humankind, betterment of humankind, and the health, and that's why current state is requiring that. One thing that I know there's a lot of researchers in academia and also in industry and national labs are working on a lot of modeling, and but even starting with the modeling the organs, right? So even before you put a more complex system like an animal, or human is even much more complex, but can you model the liver? Can you model even a cell we talked about? But I think there's a lot of progress. I've seen great models of heart, for example, human heart. But all of these things will advance, I really hope personally. But I think, it's not going to happen by just modeling or just math. But I think it requires a lot of other technologies, especially like some nanotechnologies and the specific micro sensors that actually gets the data into the model, so you can actually mimic that physics, biology, and the dynamics and chemistry. One thing maybe I quickly share, I saw at actually Northeastern University here, there's a professor working on energy harvesting microbots. So basically it's a very tiny robot. You need to give an energy to that thing to operate and run through the cells. It's actually harvesting the energy from the body heat and kind of keep going until it finishes its mission. So technologies like that, I think it keeps me a little bit optimistic, but it's a long way. I agree with Jennitte. It's not very easy, very difficult, because every animal, every human are different and unique.
Yeah, I also, totally agree. I think that I was just talking to someone earlier today. I was like, I think these AI models are hypothesis generators. We can limit the search space, but I also, and maybe you can hear this somewhat through my answer in the previous question, but I think that scientists' jobs are not going to go away. I think they're actually even more important than ever. And then there's always a lot of hype, and academic Twitter is very spicy in terms of what used and what shouldn't be used in terms of AI. I mean, some think that you can train a whole AI model based on DNA to predict proteins. I don't know if that's true, but you can create a very fancy AI model, which you can also just get from first principles as well. So I think there is a lot of excitement and trying to get to, complete, for instance, in my field, virtual cell models is going to be very ambitious and I think very worthy, but we're definitely far away from that. And even if we create that, it's still going to be not a replacement to animal models, for instance, or clinical trials.
Okay. So I saw that Walt, you put the emails in the chat, right?
Yeah, everybody on the call here has access to all of our panelists' email address. Thanks you guys for being willing to share. And then again, we're going to post our recording here on our webpage. That's the bic.ucop.edu site, so that anyone can pass this along to friends. You know, if maybe it was a tough time, midterms or test or something, you know, it's there archived for everybody to watch. So.
Yeah. And the previous ones, like we said, are there as well. So if there's topics you're interested in, you can also, you know, go and look at those. Thank you again to the panelists for agreeing to participate with this and give the different perspectives. And we'll probably bug you sometime in the future again. So, all right. Thank you, everybody.
Thank you so much. Thanks for having us.
Thanks very much, everybody.
Okay, bye.
Bye.
Good to see you, Jennitte.